Chancelloriids, to me, are one of the most interesting and controversial groups of Cambrian fossils. Are they armoured slugs? Are they sponges? Does it matter? Spoiler alert, yes, it does. The Cambrian Burgess Shale of Canada is one of the most important fossil deposits on the planet, and since its discovery in the early 1900s has been producing large numbers of incredible fossils. Many of these have proven over the years to be extremely difficult to interpret. We don't know what they're related to, we don't know how closely related they are to living animals. However, in many cases, we've started to get a better understanding of the most difficult fossils over the last 4 or 5 decades especially. But some of them are still causing problems. And among those, probably one of the best examples are the Chancelloriids. So, if you go back to Walcott's original works from 1920, you find that he included the Chancelloriids with these wonderful, strange little bits of skeleton amongst the sponges. They are part of his sponge monograph. That shouldn't be too surprising, because after all, they do look like sponges. They're tall, thin-walled, vase-shaped things, and they have this sort of skeleton of spiky bits. You might not think of sponges having a skeleton of spiky bits, but this, in fact, is a fairly normal sponge. This is the Venus flower basket. It's a deep-sea hexactinellid sponge, or glass sponge, and its skeleton is made of lots of interwoven little elements of opal called spicules. These are little spiky things, and they are what forms a skeleton in the great majority of modern sponges. However, these spicules, or sponges, come in a variety of shapes and variety of structures, but they share certain features in common. There's a limit to the range of morphology, or form, that we see in living and even definite fossil sponges. And Chancelloriids are kind of pushing the envelope a bit. Most importantly, sponge spicules, whether they're made of calcite, calcium carbonate, or opal, a form of glass, effectively, silica, they're solid. They're solid structures with, if anything, a very small amount of organic material, either as a filament in the centre or as a coating, a very thin layer on the outside. Chancelloriids, in contrast, appear to be hollow structures made almost entirely of organic matter. They are organic sort of sleeves with a space in the middle. There are some traces of a calcium carbonate crystallization on the inside of the organic material, but there's not much of it. And even worse, they're not even single elements. These are multiple structures with round bases that have all sort of joined together to make a star-like structure, which we don't see in any modern sponges. These peculiarities led a wonderful chap called Stefan Bengtsson, who sadly passed away a few years ago, to look into these much more detail. So he was taking isolated sclerites from limestone dissolution and looking at them in extreme detail in electron microscopes. And what he found there showed that there was a remarkable similarity between the structure, the microstructure of how these things were built, with that of something very, very different indeed. This amazing fossil here was discovered in the Sirius Passet biota of northern Greenland, and it is basically an armoured slug in appearance. It's called Halkieria evangelista, and this has been an iconic fossil in paleontological circles for quite a long time, since it was discovered, because it seems to show characteristics of multiple groups of organisms, all of them closely related. So the shells look like the shells of brachiopods, and these little scales all over the surface look much more like the scales that you get in other strange early creatures called Wiwaxia. And there's even similarity in the ultrastructure of these with polychaete worms. So we have annelid worms, molluscs and brachiopods, all of which are closely related modern phyla, seeming superficially at least to be united in this organism, Halkieria. And there's since been a lot more work, and the general consensus now is that this is a very primitive mollusk, but it's in this group. It's a derived animal. This is nothing like a sponge. The similarity in the structure, the microstructure of Chancelloriid sclerites and the scales, the structures on Halkieroids and another group called Sarchaetids, led Stefan and various other people, so Susanna Porter has done a lot of great work on this as well, to propose that these are in fact extremely closely related organisms, which Stefan united into a group called the Coeloscleritophila. And for many, many years this became the standard interpretation of Chancelloriids, that they may look superficially like sponges, but they're not. They're slugs related to early molluscs, which have a unique sort of microstructure of these spiny sclerites all over the surface. In recent years, though, many more extraordinary specimens of Chancelloriids have been discovered from around the world in the Cambrian rocks. They seem to be completely restricted to the Cambrian. They seem to have diversified, spread out around the world, and then by the end of the Middle Cambrian, they're gone. They just vanish. We don't know why. That's a mystery for another day. But what we do know now is a lot more about the detailed morphology. The beautiful preservation of these fossils has led to some interesting insights, which makes them look more and more like sponges. So a restudy by Stefan and Des Collins of the Burgess Shale Chancelloriids, for example, showed a remarkable number of specimens which have a clear hole at the top, an osculum in sponge terminology, which is surrounded by a little ring of spikes. And this is a feature that is not typical of something like molluscs. Beyond that, though, there's also the question of what we're not seeing. These fossils come from places like the Chenjiang biota, the Burgess Shale, the Xinjiang biota. There are all of the most extraordinary fossil deposits in which we commonly get animals, arthropods, worms with internal organs. We see the gut preservation, we see the nervous system in many cases. And not a single specimen of a Chancelloriid has ever been found with any internal organs. That's suspicious, given that molluscs should have quite a lot of them. There have also been a range of new types of Chancelloriids, in which, rather than being star-shaped sclerites with lots of rays joining at the base, what they have instead are single sclerites, open-based, rounded, but with a single axis. And this looks a bit more like a sponge spicule, possibly. It's all been coming to a head in recent years with a series of papers discussing the fine microstructure of the secretion mechanisms and whether these things have an epidermis. This is a derived feature of animals, as in it is not present in sponges, but it is in higher animals, if you like. And one recent paper this year, in fact, has led to me diving into this in more detail, which seems to have exceptionally well preserved isolated sclerites of Chancelloriids, which still have the impressions of the cells that secreted them embedded on the surfaces of the sclerites. And what they seem to show is that they had an epidermis, and therefore were not sponges. Case closed, right? They're not sponges. Therefore they are slug-like animals, mollusk relatives probably, which appear not to have had any internal organs and have a shape that is remarkably similar to sponges with radial symmetry, a hole at the top surrounded by spicules. Yeah, This is getting complicated. I think it's probably about time that we looked at ancient sponges. All of these comparisons that are being made are with effectively living sponges, of which there are 4 major groups, 4 classes, and they're all quite well understood. They're quite simple animals in the sense of not having internal organs, but they have complex biochemistry and they're highly specialised for sitting on the seafloor, basically sucking up water and filtering the yummy stuff out of it. There are exceptions. There are predatory sponges that eat shrimps and fish and things like that, but again, we'll talk about those another time. In general, sponges are relatively easy to understand, it is thought. However, when you go back in the fossil record and really look deeply into the Cambrian faunas especially, things start getting very weird indeed. This thing, for example, is called Takakawea. It's from the Burgess Shale, and it appears to be tetraradially symmetric. It has the spicules with weird blades extending from them, made of organic material, and yeah, that's just the start, really. There are so many early sponges that seem not to fit into the modern classes. They have characteristics that are quite different from anything that we see alive today. For the purposes of this talk, though, there's one group I want to focus on. These have been previously described as demosponges. They're in a lot of the literature as protomonaxenid demosponges, and demosponges are the most abundant and diverse group of sponges alive today. But there's actually almost nothing linking them together at all. The only feature which seems to have been used to support an assignment to demosponges is that it has spicules, which are basically like single needles. They're single axis, no branches, that's it. They're on a completely different scale. They have a different architecture, they have a different appearance to the sponge as a whole, a different skeletal structure. And when you look closely, there is nothing linking them to demosponges. And when you look even closer, they start getting very strange indeed, because their spicules have a very thick outer organic layer, and the skeleton inside, the mineral component of the skeleton inside, is complicated. This one is a thing called Linica that I studied over a decade ago now, and what we discovered when you had very well preserved spicules is that it has a complex internal structure which can only be explained by the presence of 2 different types of crystal. So you have an internal area which was made of silica, and that's still cryptocrystalline, so no crystals, recrystallization, and it still has a filament in the centre, which is typical of siliceous spicules. But then the outer part of it is not preserved at all, it's always dissolved in that material. However, if you go to limestones, in the Sinsk biota in Siberia, then there you find the internal part, which was silica in the Chinese material we were studying, was dissolved, and the outer part is preserved as calcite, which is staple in limestones. So what we seem to have is a spicule structure with a silica spicule in the centre, typical of things like demosponges and hexactinellids, the glass sponges, and a calcite spicule wrapped around it, which is typical of the class Calcarea. And all around the whole thing is this thick organic sheath. What this tells us is that this sponge was not like modern sponges. It's probably a much earlier branch of the sponge family tree, and it seems to have structures within the skeleton that we do not see in anything alive today. So we're going back to an ancestral state. And in these early sponges, one of the features that we see here and in many other types of these early fossil sponges is this thick organic outer layer, which might start ringing bells with Chancelloriids. From there we have to look at a close relative of Linica called Pirania, and this one is also from the Burgess Shale, but also turns up, or relatives of it later on, through the Cambrian and Ordovician and even into the Silurian. But Pirania in the Burgess Shale shows some very peculiar features. it seems to show, for example, that these spicules actually have an open base, as in Lineker, as in various other groups of related sponges. Rather than coming to a point where they are embedded in the body wall, they seem to have a flared out base which just disappears. There is no solid spicule inside it. In some of the Pyranea specimens, you even see the spicules are twisted, that these are almost like those little bag ties that you have with a strip of plastic and a bit of wire in the middle. You can twist them round and it looks just like this. So there is evidence in many of these spicules for a very thin mineral bit in the centre, and then a thick outer layer which is flexible, can be twisted, and looks as though it was mostly hollow. The point here is that we're starting to show that some of these early sponges had spicules that are much more like the spicules or the sclerites of Chancelloriia. So especially when you have Chancelloriids with only a single ray, conical things with a wide base which is an opening at the bottom, we see the same sort of things in Pyranea. We also see the same body form, this tall, thin architecture with a hole at the top surrounded by spicules. So in that way, we're starting to reconcile the discrepancies, some of the discrepancies, between sponges and Chancelloriids. Okay, but we're still left with this huge problem of there is evidence for an epidermis in the secretion of Chancelloriid sclerites. And that is a fundamental problem because sponges are just too primitive, too early a branch within animal evolution to have possessed an epidermis. It just doesn't work. Unless we've got something a little bit wrong about what sponges really are. There's one other really peculiar feature of Pyranea, which has been known since Walcott published his monograph in 1920, which is that the surface of Pyranea is covered by a sort of polygonal cellular structure in the best preserved material. It looks like an epidermis. We don't know what this structure is. All we know is that it is there in Pyranea. We don't see it in the other sponges, but it is a feature that is obvious within these fossils. As a result of these similarities between Pyranea and Chancelloriids, and the fact that they even blend into each other, I've actually published a couple of papers suggesting that these are in fact derived Pyraneids, that Chancelloriids are a member of this group of peculiar early sponges. That presupposes a few other odd things though. For example, it would tend to imply that an epidermis was there in the earliest ancestors of sponges, and in these very early primitive sponges that were branching off before the modern groups evolved, and that this was somehow lost later in the evolution of sponges. There actually is quite a lot of evidence for secondary simplification. For example, I've pointed out this tetraradial sponge, this Takakawea. There's actually quite a lot of them which have fourfold symmetry. That is not a feature of modern sponges. It's too complex an architecture. There are peculiar aspects of the genetics of sponges that imply possibly that they had some sort of precursors to muscles and or nervous systems. So there are features of the genomes which in higher animals are used in those aspects. It is also possible that the genes predate the structures that they code for. So you have to be very careful with that sort of argument. But you could also interpret this as secondary simplification of sponges from something that had an epidermis, possibly muscles, basic nervous system, that sort of structure. In which case, our idea of what the first animals looked like would have to change quite a lot. Because the last common ancestor, when you trace all of these trees of evolution back, the last common ancestor of all of the higher animals, of us, for example, and sponges, was not some amorphous blob. It was something which actually had structure, had an epidermis, was much more animal-like than we give it credit for. And that's why Chancelloriids kind of matter. Because if they do turn out to be somehow a mollusk-like animal that has evolved secondarily to become like a sponge in morphology and lose its organs and so on, then fair enough, that's a very weird branch of evolution, but it's possible. And that leaves the status quo absolutely fine. But if instead Chancelloriids are accepted as being sponges related to Piranha, and that this epidermis evidence is genuinely there, then that means we have to rethink what the last common ancestor of animals actually looked like. And it pushes back the origins of some of these features that we consider relatively complex and derived back to before that last common ancestor, which has all sorts of implications for our understanding of the very early fossil record. So there's a long way to go, but hopefully you'll understand now that these things, which look like a little dead end in evolution, something which we can look at and sort of admire from a distance and not really care too much about, can actually end up having a pivotal place in our understanding of the history of animal life. Well, I think that basically covers it for this one. For the moment anyway, until the next discovery. So in the meantime, I will wish you many happy sausage dogs and leave it there. Psst, if you made it this far, remember, hit the like button and subscribe.